Hello. Welcome to IMG's live webinar. Thank you for joining us. This session will be hosted by Chief Medical Officer Dr. Geoff Tothilll, who will be providing global updates on the coronavirus COVID-19 pandemic with a specific focus on vaccine developments. After the briefing, Dr. Tothilll will host a live Q&A session. Thank you to those of you who submitted questions during registration. We did take all those questions into account when creating the content today. At any point during the webinar, you may submit additional questions through the Q&A feature on the platform. We will answer as many questions today as time allows, and we'll follow up on any additional topics afterwards. This session is being recorded and will be available for your viewing in the upcoming days. At this time, I will welcome Dr. Tothilll to begin his presentation. Dr. Tothilll?
Thank you, Alison, and good morning, afternoon, or evening to you all, wherever you're joining us from in the world. Pleasure to have you with us. I trust you're all well. It's been a pretty difficult year, of course, globally, and we thought it would be sensible to take stock of the current situation, particularly as all the efforts to produce a vaccine seem to be coming to fruition with 3 candidate vaccines, some of which have been deployed. So in this session, I want to just pause to look at the current global situation. I want to talk about viral mutation because it's a question that's come up in the questions, but also is in the press this week in the UK and Europe, and will shortly be in the press in the US, I suspect. I'm going to briefly talk about how immunity works, how the virus infects us, how we would naturally protect ourselves from it, and how vaccines work. And then I'm going to discuss the various vaccines that are now becoming available, but with a particular emphasis on safety and try and answer some of the questions about the rapidity of development of these vaccines. There is, of course, the temptation with the vaccines coming online and people beginning to be vaccinated in both the UK and the US and elsewhere. There's a temptation to think that this virus has gone away. I think this graph, which was produced by the Financial Times from data largely from Johns Hopkins, shows that the virus is still very much an issue across the world. Interestingly, the number of confirmed cases of COVID-19 per 100,000 of the population is remarkably consistent across the world at the moment in countries seeing resurgences. And so we are seeing similar resurgence profiles in the States and across Europe. And interestingly, of course, governments are responding to these upticks in the number of cases by implementing varying degrees of lockdown over the holiday periods. So, for example, in the Netherlands, which is seeing a rapid resurgence in cases, you can see quite a steep uptick in the number of cases there. They've declared a 5-week lockdown from the 15th of December to the 19th of January. In the UK, we have a brief respite from the 23rd to the 27th of December where limited family mixing is allowed. And in France, that are just relatively flat in terms of their cases at the moment, they've lifted a lockdown that they imposed on the 15th of December. In fact, they lifted that on the 28th of November, but fairly strict measures are enforced countrywide. Germany, on the other hand, are seeing quite a rapid increase in cases, not shown on that graph, and they've closed all non-essential shops and schools during the holiday period, and that's lasting from the 16th of December to the 10th of January. So the other thing I think that's interesting, not again particularly well represented on this graph, is that lockdowns and reducing social contact certainly does help in suppressing the numbers of new cases of the virus. I want to just quickly talk about the US situation. Clearly, there are some states in which there is a rapid rise in cases. It does look statistically as if the Thanksgiving holiday did produce upticks in the numbers of cases in various states. And here I've deliberately actually picked 3 states, California, New York, and Florida, where there have been fairly large increases in the numbers of cases, a gradual climb in the number of new cases every day. I put Nebraska on there to illustrate a point about what's happening elsewhere in the states, because this would be a fairly gloomy picture if it was taken in isolation. If you look at this data, This graphic shows the trend lines in each state. And you can see that there are a large number of states where the absolute numbers of cases, or where the numbers of new cases each day are actually decreasing, as well as some key states where there's an uptick. On balance, yes, there's still an increase in number of cases per day that's being reported across the continental USA, but it is balanced by some states getting their case numbers under control. The problem with that, as I demonstrated on the last slide, is that some of these states are coming down from really very high numbers of new cases each day. And so these aren't this graphic shows trends rather than absolute numbers. So the absolute numbers are still really high. I'm going to go on and talk a little bit about this new variant of SARS-CoV-2. As you know, SARS-CoV-2 is the coronavirus variant which causes COVID. And a new variant of SARS-CoV-2 has been detected in the UK and is all over the press in Europe, actually. It's nattily named VUI 2020-12-1, which means virus under or variant under investigation, December 2012. And there are some interesting things about this particular variant, which I'm going to just quickly discuss, because it's been blown up in the press as being a very dramatic and very serious development. But I want to put that in perspective somewhat in the next couple of slides. One thing that maybe is significant is that the genetic changes in the virus that have been detected in this variant have been seen in the code that produces the spike protein on the surface of the virus. And I'm going to talk quite a lot about spike proteins in the next half an hour or so. They are very important. They're the proteins that are expressed on the surface of the virus, and it gives it the ability to get into our cells. It's also the part of the virus that we detect in our testing systems. So should we really be worried about this new virus, this strain, if you like? So interestingly, the new strain has been seen in the UK in areas of rapidly rising case numbers. So that has raised the possibility that this new virus strain is more easily transmitted from person to person. The jury's out on that. We really don't know yet. It could be that we're just testing more in places where there are lots of cases already. And it could also be that that's the place where this new variant has arisen, and so we're seeing large numbers in those areas. We will begin to get a feel for whether this new variant will predominate in the next month or so. However, on the other side, to balance that, we know that there are that other human coronaviruses that produce common cold-like symptoms, they commonly change. They change regularly, their genetic makeup changes without significant change in the severity of the disease or their ability to spread. On the concerning side, this variant has 17 specific mutations. We've seen almost all of those mutations elsewhere in the world already. But this is slightly different in that we've seen all of these 17 mutations all in combination in this particular variant. That's not been seen before. Except, of course, in coronaviruses that infect animals, we do see a similar type of genetic shifting. And we've had to produce vaccines for animals which are polyvalent. In other words, they are able to protect against a whole range of different variants of the virus. On the concerning side, it's thought to affect the spike protein, and that's important, and I'll go on to talk about the spike protein again in the next slide. But what's less known is that this virus is continually mutating, and there have already been up to 4,000 variants of spike protein identified already, none of which are thought to affect either transmissibility or the severity of the disease or the ability of the vaccine to protect. But that is the worry. The worry is that there will be such significant change in the spike protein that the structure of the virus will change somewhat, and it will become less susceptible to the immune response that we raise as a result of being vaccinated. So there's just the potential for both the COVID PCR antigen test and the vaccine to be less effective. Although, again, we don't think that that's going to be the case. On the hopeful side, the new technologies, particularly RNA vaccine production, allow rapid re-engineering of our vaccines so we can retarget the altered surface antigen if that becomes necessary. we've got to remember that every year we produce new flu vaccines because the genetic makeup of the flu virus that circulates every year changes. And within those vaccines, they're polyvalent as well. So we're well used to the concept and the technologies involved in producing new vaccines. What the recent research has done is it's made it a lot easier for us to produce those vaccines quickly. Does it cause more severe disease? Again, there's no evidence at the moment that this new variant will cause more severe disease. And no evidence, in fact, that any of the variants have produced more severe disease. Because the virus regularly changes its genetic makeup, virologists and epidemiologists use that genetic change to be able to track outbreaks in particular countries. And that's been how we've been able to tell how the virus has been imported from one country to another. So I just wanted to talk, before I go on talk about vaccines, a brief introduction to how the SARS-CoV-2 virus gets into our cells. Here's a diagrammatic representation of the virus, and the blue pins sticking out from it are these the so-called protein spike. They are surface structures around the side, on the surface of the virus, which have the ability to link into, they need to be activated by another protein, but essentially they dock onto the angiotensin converting enzyme receptor, which is on the surface of human host cells. Those receptors are found in most cells, actually, but predominate in our pulmonary parenchyma. So in other words, our lung, the surface of our lungs and upper respiratory tract. And so that's why the virus is able to gain a foothold, because we breathe it in, either in droplets or we get it onto our mucous membranes. The spike protein attaches to the ACE2 receptor, and the whole virus is encapsulated in the cells, enters the cell. So here's how the virus replicates. Top left, you see the coronavirus with its own genetic material. It's an RNA virus, so it has RNA which can code for a complete new version, a complete new copy of itself within the viral capsule. The spike protein locks onto the ACE receptor. The virus is pulled into the cell, internalized into the cell, where it breaks down. It uses the cell's own manufacturing capability to take its RNA and turn its RNA, if you like, into a copy of proteins, and the proteins are rebuilt into a new copy of the virus, and then the virus passes out through the cell wall and is released into the surrounding tissues and the bloodstream. And that's how a coronavirus replicates in our cells. So the spike protein is really critical in this process because it allows the virus to gain access to our cells. The spike protein also has a role in producing antibodies. And so this is just a brief overview of how we would normally produce immunity to illnesses. If you have a pathogenic particle, either a virus or a bacteria, something foreign that gets into the body very quickly recognizes that as being foreign or other, and it produces antibodies to that, what we call antigen. On the surface of these invading organisms, there are specific proteins, like the spike protein. The body recognizes them and produces antibody. It also produces things called T cells and other forms of immunity. And those antibodies lock on to the invading particle. And you can see measles top left and the flu virus bottom left. Chickenpox is shown. And those antibodies prevent the virus from getting into the cells. If you haven't ever seen that particular pathological particle, that particular virus or bacteria, you don't have antibodies to it. So the body takes time to develop the antibodies, if you like, learn that virus is foreign, and then it mounts an immune response, and you produce antibodies. And those antibodies persist for a while, sometimes for a very long period, sometimes for a short period. But in your immune system, you usually carry the memory of that pathogen. So if you see it again, if you get infected again, your antibody response is much quicker, and you clear the virus or the bacteria from your system much quicker. And so, of course, the the SARS-CoV-2, the coronavirus that causes COVID, we'd not seen as a species before. So we were what's called immunologically naive to it. None of us had seen quite that sort of virus before. So we had no antibodies to it, and that's why it spread so quickly throughout our communities. I'm going to move on to vaccines. One that clearly we've developed vaccines to the coronavirus very much more quickly than historically we've been able to. It's often said that it would take up to a decade to take a vaccine from conceptualization to market, whereas we've done this in a year, or under a year, in fact, in the case of coronavirus. I just wanted to answer the question, which is, how have we been able to do that safely? I think a couple of things have contributed to that. Firstly, we've known for a long time, vaccinologists have known for a long time, that we needed a system for rapidly producing vaccines when viruses change their makeup. So if we had varying strains, which we discussed before. So we've been looking at our RNA vaccines and other technologies to replace the rather more old-fashioned technologies, so we can rapidly change the vaccine that we want to deliver and rapidly produce it. That's the first thing. The second thing is we've known a lot about coronaviruses. We've seen SARS, of course, was the first serious pandemic coronavirus that we saw. And then MERS was also studied extensively, and we had candidate vaccines for both of those pandemics. So we've known a lot about coronaviruses, and that's why we've been able to had a flying start, really. We've also known about the spike protein, the structure of the spike protein for coronaviruses. So again, we had a good target to look for when we were developing vaccines. Of course, secondly, the amount of resources that we've thrown globally at producing a vaccine has been enormous. And thirdly, we've been much better at recruiting people to studies. The efficiency with which we can recruit people to studies and report on those studies has improved hugely in the last couple of years. So all of those things have come together really in the last 12 months to enable us to produce these vaccines very quickly. Secondly, there's a question of safety. And I wanted to just explain why the vaccines we're seeing for coronavirus are so safe. Previously, we used to use various ways of vaccinating against disease. One was that we would give a weakened version of the virus that we were protecting against, or an inactivated form of the virus. Now, clearly, there is more of a chance with immunocompromised patients, if you're actually giving them a weakened form of the virus, that they will get sick as a result of being given that weakened or attenuated virus. And so there is some possibility of reactivation of these vaccines, although it's incredibly rare. And even with vaccines that are either weakened virus or attenuated virus or inactivated virus, we see very, very good safety profiles. But the new vaccines don't depend on giving any sort of attenuated or inactivated virus to the patient. The Oxford Astrazeneca vaccine candidate uses a harmless virus which causes a cold-like illness in chimpanzees, but it doesn't cause any illness in humans. And that virus as a vector, as a way of getting the genetic code into our bodies and to produce antibodies, that virus is harmless to humans and doesn't replicate in our bodies. It's been genetically altered, so it doesn't replicate in our bodies anyway. So what you do is you take the gene for the spike protein, you incorporate it into this harmless viral genome, you inject that into the body, the virus is taken up by our cells, and our cells produce the spike peptide, the spike protein, and that's released into our bloodstream. And then our body produces antibodies to the spike protein. So if we then see the spike protein on a coronavirus, we automatically have those antibodies in our bloodstream, and therefore we mount a defense against the coronavirus. It's an extremely safe way. It's a tested way of delivering the spike protein genetic material to our cells and producing an antibody response. A much more modern way, if you like, a more advanced way of doing this has been developed by Moderna and others. What happens here essentially is you take the code for the spike protein, the RNA code, you introduce that directly into the nucleus of our own cells, and then our cells are prompted to generate the spike protein and release that into the bloodstream. And that's on the right side of that. I'm not going to discuss DNA vaccination because it's slightly more complex, but RNA vaccination. The advantage of RNA vaccination is we can change the makeup of our vaccine very quickly. So if the spike protein changes, essentially what you just need to do is change the RNA sequence that you inject as part of the vaccine, and your body produces antibodies to the new spike protein. So in theory, each year, or however often we need to do it, we should be able to generate new vaccines for each variant of the virus. We really don't know whether we're going to see seasonal variations, but if we do, this new RNA technology should make it much quicker for us to produce new vaccines. The other thing about RNA vaccination is it's inherently quite safe, but very safe, because the RNA that you inject in as part of the vaccine breaks down after having coded for the viral proteins, the spike proteins. The RNA is destroyed in our cells through a natural process, which is part of a normal cycle of cell metabolism. So it's an extremely safe and effective way of producing immunity. So what do we mean by vaccine efficacy? So essentially what you do when you test vaccines is you give the vaccine you split your group of volunteers, if you like, into two groups. One group gets the vaccine and the other group gets a placebo. They're blinded to that. In other words, neither the patients or the people, or rather the volunteers, or the people administering the vaccine know whether they are giving a placebo or the real vaccine. And then what you do is you watch each group and you see how many of each group develop the disease. And the vaccine efficacy, which is, if you like, the trial result, is interpreted as the proportionate reduction in disease among the vaccinated group. And as I say here on this slide, so an efficacy of 90% indicates that there's a 90% reduction in disease occurrence amongst the vaccinated group. Or if you like, a 90% reduction from the number of cases you would expect if they'd not been vaccinated. I suppose it's important to make a distinction between efficacy and effectiveness. Efficacy is the result that you get through the clinical studies that are being conducted. Effectiveness is a real world, if you like, a real world measure of how well the vaccine protects you. And we usually expect the effectiveness of the vaccine to be slightly less than the efficacy seen in trials. So how effective are the vaccine candidates? As you can see, the Moderna, the Pfizer, and it's claimed, although we haven't seen greater scientific evidence for it, the Russian Sputnik vaccine are all shown to have at least above 90% efficacy. The Oxford vaccine initially reported 70% efficacy. There is some indication that giving a reduced second dose produces efficacies above 90% as well. And we're just waiting for the final results of further trials. Just want a quick note. Usually, we would see a reduction in the response to vaccines amongst the elderly. And of course, it really is the elderly we're trying to protect. Of all the risk factors that make you more vulnerable to COVID-19, age is the most significant risk factor still across the world. And the older we get, the poorer our response to vaccination and the less effective our immune response. Although we are seeing actually with all of these candidate vaccines, good immune response even in the elderly. So that's very hopeful. How long that immune response will last is yet to be seen. I just want to talk about safety briefly. In medicine, there is an important difference between safe and harmless. And I think if you mean that there's absolutely no adverse effect possible, then no vaccine is completely safe and no drug is completely safe if what you're looking for is absolutely no chance of any adverse effect. And that quote's from Professor Stephen Evans at the London School of Hygiene and Tropical Medicine. I think he's absolutely right. Even the most benign pharmaceutical has a risk profile. And what we really mean by safe is, what's the balance between the unwanted effects of that drug or vaccination compared with the benefit? And if it's very clearly beneficial, then we would describe that as a safe intervention. And of course, safe is a relative concept. There are lots of chemotherapeutic drugs which can do some really unpleasant things to you, but balanced against the risk of recurrence or progression of a cancer, for example, we would describe those as relatively safe. Vaccines, by this measure, are extremely safe. And all of the vaccines that are reported phase 3 trial results have been shown to be safe with this as a criteria. So just quickly, where we are with vaccines, we know the Astrazeneca Oxford vaccine is effective, particularly in the altered dosage regime. It does protect from severe disease, and it's inexpensive. The Pfizer and BioNTech RNA vaccine has been shown to be effective both in the elderly and other age groups. It has the disadvantage of requiring long-term storage at very low temperatures, but it's already being deployed and given to people in both the US and the UK. And the Moderna vaccine has the advantage of being more stable at higher temperatures. Again, it's been shown to be highly effective in all age groups, protects from severe disease, and it's moving very close to approval by the FDA. I think it was approved by a specialist committee at the FDA a couple of days ago. So we are looking forward to that being licensed in the US shortly. A quick recap on therapeutics. I'm not going to spend too much time over this. Overwhelmingly, the therapeutic intervention which is proven most effective to reduce mortality, i.e. deaths from COVID, has been the giving of steroids, dexamethasone. It's especially effective for patients on ventilators or receiving supplemental oxygen. And indeed, that's the standard of care, really, that it is given to patients requiring supplemental oxygen as a result of COVID-19 disease. A lot of interest now in Regeneron's antibodies and other antibody, monoclonal antibody therapies. We know that patients that receive the Regeneron antibody cocktail had far fewer medical visits related to COVID-19, and it's certainly been advised for use in the early stages of illness. And then remdesivir, the jury still is out on remdesivir. It shows some promise as a therapy that improves time to clinical improvement and discharge, but doesn't seem to hugely affect mortality. The WHO Solidarity trial, which was published recently, didn't show that it reduced mortality or the need for ventilation or duration of stay. So there are rather a lot of conflicting papers around on that at the moment. I just want to re-emphasize the safety and usefulness of vaccination, really. That's just a slide showing vaccine preventable diseases from 2009. There are some to add to that. But as you can see, there's a huge number of diseases that we can prevent now or reduce the severity of by using vaccines. And this is a fairly telling slide as well. Sorry, it's not terribly readable, but on the left, you can see the pre-vaccine era morbidity in the US. So that's not mortality, that's morbidity. Those are people infected with have clinically significant episodes from the various diseases. And on the right is a comparison, a recent comparison actually from this year. And as you can see, the reduction in the morbidity from all of these diseases as a result of vaccination is enormous, and 100% reduction in morbidity from diphtheria. And you can see on the slide, it's an extremely effective way of reducing the burden on our healthcare systems, but also the burden of disease in the population. So just in summary, what do we know about the three leading vaccines? We know that on a population scale, the risk to an individual of having the vaccine is extremely small. And the risk is outweighed by, in all age groups, against when measured against the risk of getting COVID-19. So the risks of having the vaccine are less than the risks of catching COVID-19. They all produce good neutralizing antibodies and T-cell responses, and they protect well against the current strain of COVID-19. Whether they will successfully protect against all the strains as the virus mutates, we don't know. But the indications are at the moment that they will almost certainly protect against the strain that's around the world at the moment in all its variants, and probably for up to a year, although that's rather speculative. You need 2 doses for all 3 vaccines to be effective, and elderly patients raise a good antibody response. What we don't know how long the antibody response lasts. We will get a better impression of that as the months go by. We don't know if a reduction in antibodies means less immunity and protection. You would think that it would, but that's not always the case. It is quite possible to have low levels of circulating antibodies, but actually mount a robust defense against the virus when you see the virus again. We don't know whether vaccination will significantly reduce infection rates. If a vaccine, and there are some vaccines that do this, but if a vaccine completely eliminates the virus from your body, or at least detectable virus, and therefore you don't spread the virus, so you're not shedding the virus, then that's called sterilizing immunity. We don't know whether the COVID-19 vaccines will produce sterilizing immunity. It would be highly valuable if they did, but we don't. The jury's out on that. So we don't know whether, once you've been vaccinated, whether you can still acquire and shed the virus. What we do know is that you're very unlikely to get severe disease. We don't know if booster doses will be required. We talked about changing the virus makeup, so we don't know what the effect of that will be. And herd immunity is another much discussed topic. And people quote percentages of the population who would need to be immune between sort of 50 and 70%. So it does mean that the vaccine uptake will have to be considerable. The other problem with, of course, herd immunity is it's herd immunity to the vaccinated, to the virus that you're vaccinating against. If this virus changes its makeup, its genetic makeup, herd immunity doesn't apply anymore, because the herd immunity you've acquired will be to a previous, if you like, version of the virus. And so herd immunity may not apply at all in this case. So herd immunity, for example, to flu doesn't exist because flu virus changes every year. I just wanted to end on a festive note. I think we've all been extremely, I think we have a debt of gratitude to Johns Hopkins, actually, who have kept us impartially appraised of the status of the data on the spread of this virus, and latterly on the deployment of vaccines. And I was reading one of their articles about the deployment of the vaccine in the US today, and I came across this. There are unverified rumors that Santa Claus and his elves may be among the essential workers prioritized to receive the first COVID-19 vaccine doses. I thought that was rather lovely. Somebody at the end of a very grim year in Johns Hopkins has a sense of humor, and that was buried in a scientific paper that I read. So it really just leaves me to wish you all a very happy holiday period. I hope you and your families and loved ones stay well. And thank you for joining us for this series of webinars. I hope they've been helpful and supportive. And of course, I'm quite happy to take questions now.
Great. Thank you so much, Dr. Tothilll. So we're going to jump right in here to the live Q&A. We've had some questions trickle through during the session, so we'll get as many done as we can here in the next 10 minutes or so. So I'm going to jump right in here. So our first question is, if somebody has already had COVID, should they still receive a vaccine?
Yeah, that's a very good question. The answer to that is yes. The consensus opinion is that you should be vaccinated. And the reason for that is that natural immunity that you get from having had the virus is thought probably to be weaker than the immunity that will be derived from having the vaccination. There doesn't seem to be any reason clinically why you shouldn't have the vaccination. It won't do you any harm. If anything, it'll boost your immune system and give you a longer protection than you would have got naturally from just having COVID. So the answer to that is yes.
Okay, great. The next one here, there are quite a few vaccines, as you have walked us through today, that are available now or going to be available. So what's the thought on receiving more than one vaccine?
Yes, that's also a very good question. So in general, the feeling is that you should, and the way this is being structured in both the US and the UK and elsewhere, is that you will get the booster dose of the vaccine. You'll have the same booster dose, so the same second dose, if you like, because I'd say they're two-dose regimes. You will have the second dose, and it will be the same type of vaccine, the same manufacturer, the same delivery mechanism as the first one. And the reason for that is we don't have a lot of data on what would happen if we mix and match the vaccines. So if you're having the Pfizer vaccine, then you will be booked in for your second dose, and it'll be the Pfizer vaccine that you'll get. If you get the Astrazeneca one, again, you'll get the second dose of Astrazeneca vaccine. In other areas of vaccinology, and certainly with things like the rabies vaccine and other vaccines, if we have doubt about vaccination in emergency situations, we have used different types of vaccines against the same disease in the same patient. But we have absolutely no data for these vaccines. And so erring on the side of caution, the approach will be that you will stick to one sort of vaccine. Whether that will change in time, difficult to predict. But certainly in the first 6 months of this, as we're learning more about the vaccines, you'd be well advised to stay with a single type of vaccine.
Great, thank you. I'm going to shift gears here a little bit and get to a couple questions around travel. I think that's on a lot of our minds here on the webinar today. So one question that we have here is, do you think that passengers will be required to present documentation that they've been vaccinated prior to being allowed to travel?
Yeah, that's a very interesting question. Earlier in the year, we all speculated that one of the solutions when vaccines were available, one thing that would unlock our international travel and to a certain extent our economies, would be a vaccination certificate. Now, there are difficulties with that, although not insurmountable, but really sort of administrative difficulties. One is that there will have to be an internationally accepted mechanism for documenting, recording and documenting everybody's vaccine status. So there'll have to be a common certification, if you like, which is recognized across the globe, which will give the countries confidence that their inbound travelers have actually legitimately been vaccinated. Now, we've developed this across the world for yellow fever, of course. For many years, we've had countries that demand yellow fever vaccination certificates for inbound travelers coming from areas where it's endemic. And that's something you could see developing. The problem with that is that I suspect it'll take a long time for nation states to agree on the protocols and the requirements and the documentation required. Theoretically, once we know the duration of cover from the vaccine, this could be seen. And I think probably we'll see it arise if we see a significant antigenic shift in the virus. So if the virus stays static and we know that our vaccinations globally are effective, then within 9 months to a year, we'll have vaccinated a significant percentage of the world's population, maybe 18 months. And beyond that, if there's no antigenic shift, we'll know that and there's persistence and the immune response, then there won't be a need for vaccination certificates, because we will assume that the majority of the world is vaccinated and travelers will be sort of resistant to it. However, if we see antigenic shift like we have in the flu viruses, then I suspect current vaccination certificates will be one mechanism where we can stop this virus from spreading around the world. So I think there are lots of variables leading up to those decisions, but I suspect that Q1, Q2 next year, it'll be very much on governments' minds as to how they might implement such a process. I'm not entirely optimistic we'll see it next year, actually, now.
So on the same, I think on the same topic here, focusing on travel, there are a couple of questions around, let's say, inconsistencies that we're seeing across the globe for both quarantine requirements and testing. So I guess a two-part question here is, why do you think that we're seeing that variance from country to country? And also, do you foresee that in the upcoming months, we have a more consistent process and method for individuals arriving into a country with regard to quarantine and testing?
I would love to be optimistic and say that I can see the world coming together and having a single approach to quarantine and testing. The truth of it is that lots of governments put process into place as a response to political pressures as much as anything. And in countries where there are rapidly increasing case numbers and a feeling in the population that governments aren't doing enough, sometimes quite draconian quarantine measures can be put in place without necessarily the science that shows that they're effective. So I think we will continue to see divergence of countries across the world, and they will respond as they are in terms of local lockdowns as well. Actually, they'll respond to the trends. The trends in their case numbers within their countries, as much as the risk posed by inbound travellers. So I'm not optimistic that we'll see consistency. That's the first thing. The second thing about I think increasingly we'll begin to understand how we should effectively use testing. The lateral flow test, the antigen test, rapid antigen testing that's been developed, although not quite as effective at detecting cases as PCR testing, in some cases is proving really quite respectable as a rapid assessment of whether you got the virus or not. And I think we will start to see countries accepting certain sorts of lateral flow tests, rapid antigen tests, as a requirement for entry. The problem with the rapid tests is they're often done outside clinical environments. And those tests can be done by individuals, they can be done randomly in airports. They can be, which is helpful to the patient, or rather the person being tested, but means that they are less authoritative sometimes as PCR testing done in a lab. And I think that's why we're seeing a lag in countries accepting rapid antigen testing as being suitable for passengers inbound. I think that will change. I think over the next sort of 3 or 4 months, we'll see more countries accepting rapid testing. But I think that rapid testing will probably be done at airports. So there's some surety that those rapid tests are accurate and have actually happened, if you know what I mean. So there's two parts of it. Is the test accurate and reliable? Is the false positive and negative rate acceptable? And can we demonstrate reliably that they have actually been done and they're not sort of just invented by the passenger for convenience? So I know that's a roundabout answer, but I hope that some of that's helpful.
No, certainly very helpful. Okay, I think we can do one more quick question here in the last 2 minutes. So we have a question here. You had a slide around vaccine efficacy, and you described the difference between efficacy and effectiveness. So the question here was, how much of an incidence of COVID-19 in a population needs to be present to determine the vaccine effectiveness?
That's a very good question. I think the answer, at a population level, I would struggle to answer it. But at a study level, I think I put just trying to find it now. I mean, we are looking at sort of, I think we're looking at about 20,000 people on the Pfizer trial, 15,000 on the Moderna vaccine, 10 to 20,000 in the Oxford Astrazeneca trials. Those are from a regulatory standpoint, those are ample numbers. And so really, we've already hit numbers where we know that the vaccine is effective. Effectiveness and there is a difference, of course, in efficacy and effectiveness, but with other vaccines we don't see huge differences. And so I would expect that, within a month of starting our vaccination programs in the US and the UK, we'll be able to get some sort of indication of effectiveness. I don't expect it to be vastly different from the efficacy rate actually in these vaccines. I think, in other words, I'm not expecting it to be sort of 50% less. I expect it to be sort of 80 to 90%. And the other thing, of course, is some of these studies will not have caught everybody who has, even in the vaccinated group, they will not have been able to identify everybody that came in contact with and had the virus in their throats, for example, in their respiratory tract. So it does become quite difficult to be absolutely sure what the effectiveness is in a population unless you swab everybody all the time. But certainly, 90% is what we'd expect, around 90%.
Great. Thank you again, Dr. Tothill. This does conclude today's webinar. This entire session will be available to view on demand in the upcoming days. You will receive a link by email if you registered today. And we also welcome you to visit our website for the latest information on COVID-19. And we will be sharing details on future webinars, which we welcome you to join as this situation progresses. Thank you all so much for joining us again today. And from all of us here at IMG, we wish you and your families a happy holiday and New Year. Thank you.